Hi everyone, my name is Maël Gaonach and I'm gonna introduce you to py-ISPyB, which is a new implementation of a LIMS for experimental structural biology. It's a new implementation because there was already one existing implementation, which is ISPyB. So I'm gonna start by presenting what it is and what it does. You've heard of it before this week, but let's go to a more in-depth introduction to it. So ISPyB means Information System for Protein Crystallography beamlines. Basically, it's a database that handles a lot of different data along the process of structural biology experiments. So we have data coming in before the experiment first of all, before the experiment on this side. So the first thing coming to ISPyB is the sample definition when the users submit the shipments that they will send to the facility. And then we have the sample management part of the database. So this handles the definition of what is inside the sample, which is crystals and proteins and how they will be placed on the sample changer on the beamline. So all the data comes from the user portal that runs at the facility. And then ISPyB comes also during the experiment. On this side, we handle a lot of information about the data collection to monitor how they are going. So we will get the diffraction images, the crystal snapshots to see what the crystal looks like on the beamline during the experiment, and then some processing outputs from the processing pipelines. So this works in connection with the beamline control module and with the auto processing workflows. And the third step is the data coming after the experiment. So this will allow to retrieve all the information about the experiment after the experiment to make sure to allow better processing and to enable the user to have a memory of what they did with what parameters. So we get all the sample information, the beamline parameters that were in place during the experiment, and also we get some information about the processing results. And from all this information, we can create PDF experiment reports for the users, and also we can make exports of all this information to the home laboratory. So here is what the ISPyB system looks like at ESRF. So first of all, we got a list of beamlines connected to the system. All these beamlines will produce the information, and this information will be uploaded by the beamline control system to the ISPyB API. This information is completed by the user portal information about the proposal, about the samples, etc. And then we can visualize all the data in the ISPyB database through graphical interfaces. So at ESRF, we have two of them running. We have the legacy ISPyB interface, which is implemented in Java, same as the ISPyB API, which is the Java 2. And then we have a JavaScript GUI that is currently in use. So the system that I showed before is the one running at ESRF, but ISPyB is a collaboration across many facilities. So you can see all of them here. So they have different installation going on, but it's the same principle behind. And now about the new implementation. So the new implementation is py-ISPyB. The goal behind this new implementation is to start a new collaboration with the same facilities, but this time to start a full stack collaboration, which means that we will develop a new API to go on the same database schema, and also develop a new user interface to go along with it. So on the server side, the API, we started a technical redesign to allow using newer technologies, because the currently in use API has been developed over the last 20 years. So a big part of it is aging, and it needs to be redesigned with the new technologies to make maintenance much easier. So we decided to change the implementation language from Java to Python to allow much better maintainability, because across all the facilities using ISPyB, Python is the main language used in most applications. So we have a lot of competences for that. And then we decided to go for a more modern API, which is using the REST principles, whereas the Java API was using the SOAP API principle. And then we decided to include new administrative and scientific requirements. This includes, for instance, the new types of proposals that have to be handled by the system, which are the BioSAXS proposals. And we decided to have a backward compatibility with other existing tools connecting to ESPyB, which can be user portal, beamline control module, and also graphical interfaces. This makes the transition to the new server side easier, because we don't have to redevelop everything that goes with it. And on the user interface side, we decided to use the latest front-end technologies. So we went for React, and we developed it so that it is easy to extend. And as I said before, we decided to include it in the collaboration so that everything is in the same package. And same as for the server side, we went for a backward compatibility with the currently in use API, but this only works with limited functionalities, because of course the new functionalities will only be implemented in the new API, so they will not be available in the new user interface when it's connected to the old API. So how do we organize this collaboration? So we split the work in a lot of different working groups. For each of those, we have a responsible facility, and then some other participant facilities. I will present you now the progress that was made so far from the different working groups. So first, so the how we organize the collaboration with the working groups is that every week we have a coordination meeting with all the working groups together to report the decisions and progress from the different working groups. And then we do the different technical discussions that are specific to the different working groups through GitHub issues, and also by creating working group dedicated meetings when it is necessary. So on the framework and architecture working group, we set up this, this framework for the project. So as I said before, first of all, ISPyB is a database. So we created a repository for the database schema. And this is used as the source of truth for the database schema, and all the other repositories will use it. So when this schema is updated, we have set up some GitHub action pipelines to update some Python models that are connected to, that allow Python to connect to the database. And we also have set up a Docker container containing a pre-populated database that can be used for testing, so that all facilities can run their test against the same data set. So as I said before, the API is connecting to the database using the model, and then the UI connects to the API, and this allows the user to see what's inside the database. On the user portal synchronization group, we have developed some generic way of synchronizing the user portal from the different facilities which run different user portal software. To do that, we have developed a generic input format that is created at each facility. They will generate the same input format and provide it to py-ISPyB so that it can create the relevant entities in the database, which are proposal, session, proteins, samples, and persons. On the authentication and authorization side, we have developed some plugin system for the authentication. So we natively support LDAP, Keycloak, and a development authentication, but we also allow to add any other authentication that the different sites might be using. And we have developed a system to allow authorization to the data through different authorization levels. So we can authorize a user for the whole proposal, or for individual sessions, or for all the sessions going on in a specific beamline. So the first use case for this is the development of serial synchrotron crystallography at ESRF. So I will show you what the UI looks like for this use case. This is still under development, so for now it is filled with randomly generated test data. So don't look at the individual data, but more the structure of the UI. Jerome already introduced you to SSX, but the important thing here to know is that instead of having one crystal and many images, like in MX, which was the first purpose of ISPyB, we end up with many crystals and one image per crystal. So of course, the whole data schema will be different. So we had to modify the UI and the database to handle that. That was already presented. So on the new UI side, we now have a list of, this is a list of the different experiments that were run in a beamline session. So we can see that we have a different, compared to ISPyB, we have a different sample definition. We can define the sample composition in a more detailed way. Then when we we can click on this button to monitor a specific experiment. And we will get this view that will allow the user to monitor the experiment when it is happening and to get feedback on the data collection that they make. So each line here will be a data collection. And we can see that we have feedback on the images that were taken and the number of crystal hits and indexed images. And then we get some other parameters about the data collection. For the future development, we need to improve the serial crystallography implementation because this is still a work in progress. And we need to switch all the existing techniques from ISPyB, the previous ISPyB UI to the new one, and same for the backend. Thank you very much.
Questions from Maël? I have one. So what makes your decision to use fast API instead of Flask, or I mean, because there are different technologies, and I know that some ESRF projects use Flask.
Yes.
Why?
So we analyzed the different possibilities for creating a API using Python. We saw that there was other possibilities like Flask. The conclusion was that they are all very good solutions, and we chose to use Fast API because it is a all-in-one solution. We get lots of different very useful features like data validation and documentation, and everything is already included, so that's less development to do to make everything work together.
Are there other questions? So, yeah. So, how far does it go in displaying the results? Does it go up to basic molecular replacement?
Up to what?
Molecular replacement, for instance?
No. So here we don't see the actual result. We see the metadata, like all the experiment parameters, and the actual result will be displayed somewhere else. This is only to have a memory of what the parameters were when the experiment was run, and all the information on the sample and everything that goes around.
Okay, then. Oh, last quick question.
So there was the legacy ISPyB webpage that existed for probably 20 years, right? Then you had EXE, but presumably you're still using the old ISPyB webpage because not everything you wanted ended up in EXE. How are you going to prevent your new web interface to ISPyB being a third one that means that you're still missing features from EXE and also missing features that people still require the original ISPyB?
So for now, we are only focusing on SSX. So the other features still are on EXE on the other one, and will still be used in this one for now. And in the future, we plan to convert everything into the new interface. But
it's actually how are you going to make sure that happens?
Lessons from the past, maybe. I think now they are warned about that. Okay, maybe this is a discussion we can have afterwards. So, well, thank you, Maël, for your presentation.